19. From where it all stems. In the late 1950s, treatment of leukemias and other cancers of the blood involved radiation to kill cancerous blood cells and subsequent bone marrow transplantation to replace them. It was known that bone marrow transplants, a procedure then in its infancy, replenished the essential cells of the blood system, but there was no understanding of the source of these cells. The three different types of blood cells had been identified in the late 19th century: red blood cells, which transport oxygen throughout the body; white blood cells, which protect against germs; and platelets, which keep us from bleeding. It was known that they came from inside the bones, in the bone marrow, where trillions are made every day. Immature forms could even be identified. But how they were produced was a great mystery. Meanwhile, outside the laboratories, the Cold War was raging, and many people feared a nuclear war. Researchers were looking for ways to treat people, most likely military personnel, who might be exposed to whole-body irradiation from nuclear weapons. Ernest McCulloch, a physician, and James Till, a biophysicist, at the Ontario Cancer Institute in Toronto, were part of this effort. They set out to measure the radiation sensitivity of bone marrow cells and to determine how many bone marrow cells were needed to restore blood cell production in irradiated mice. The design of their study called for a group of the irradiated mice to be killed 10 days after transplantation of various numbers of marrow cells. Normally, on a weekday, a lab technician would be the one to dispatch the animals, cut them open, collect the specimens, and hand them over to the scientists. As luck would have it, in 1959, day 10 fell on a Sunday. McCulloch himself, not a laboratory technician, came in that afternoon to do the dirty work. When he opened up his irradiated mice that Sunday, McCulloch was intrigued to find nodules on the surface of their spleens. This observation, which easily could have been overlooked, revealed changes that would have remained undetected within the bone marrow except under the most meticulous examination. The nodules were foci of new blood cell formation. Blood cell formation in the bone marrow, sequestered deep within the cavities of the bone, occurs among its interweaving lattice of supporting connective tissue only at specific scattered sites. In the mouse, however, blood cell formation occurs also in the spleen. The observation itself was thus not very surprising. What was surprising was that individual nodules contained dividing cells, some of which were differentiating into the three main types of blood cells: red cells, white cells, and platelets. This finding meant that something capable of making all blood cell types, a blood-forming stem cell, was trapped in the spleen. Moreover, it was as rare as Waldo among the transplanted marrow cells, since as many as 10,000 of these had to be injected for each nodule observed. Establishing an accurate ratio between the number of marrow cells transplanted and the number of nodules observed suggested the possibility of a single formative source. But how to prove it? The nodules had newly formed blood cells, not only of the various types, but also of varying stages of maturity. Where did these colonies stem from? McCulloch and Till would have to work backwards, like tracing a family tree back to the primary ancestor. And then fortune smiled. With the assistance of Andrew Becker, a graduate student, they devised an ingenious and elegant experiment using mutated cells that arose from irradiated bone marrow. By documenting that the blood cells in each spleen nodule bore a particular genetic signature, the investigators proved that diverse blood cells come from individual stem cells. Since there is no specific feature that allows stem cells to be identified by microscopy, this discovery was a huge conceptual achievement. McCulloch proudly called this hematology without a microscope. The concept of stem cells had been around since at least the early 1900s, but efforts to find them using the light microscope and histological stains had failed because the stem cells were so few in number and indistinguishable from other blood cells by appearance alone. Said Till, "Our work changed the emphasis from form to function. We stumbled across a way of looking at the developmental potential of stem cells by looking at the descendants they could give rise to." McCulloch and Till established the two main properties of stem cells: self-renewal and differentiation into specialized cells that have limited lifespans. Genes in the stem cells and factors in the tissue environment are important in promoting normal stem cell duplication and specialization. When a stem cell divides, each new cell has the potential to either renew itself for long periods through continued cell division or, under certain physiologic or experimental conditions, become another type of cell with a more specialized function. Unlike embryonic stem cells, which are defined by their origin in a 3 to 5 day old embryo called a blastocyst, adult stem cells in mature tissues are of unknown origin. The adult tissues in humans that are now believed to contain stem cells include bone marrow, blood, brain, blood vessels, skeletal muscle, skin, and liver. There is a very small number of stem cells in each tissue, where they may remain dormant, non-dividing, for many years until they are activated by disease or tissue injury. The discovery of stem cells has opened up the exciting possibility of cell-based therapies to treat disease. Some examples of potential treatments include replacing the dopamine-producing cells in the brains of Parkinson's patients, developing insulin-producing cells for type 1 diabetes, and repairing damaged heart muscle following a heart attack with cardiac muscle cells. But there is a dark side to stem cells, one that offers a fascinating line of study for researchers and a ray of hope for the human race. Growing evidence suggests that stem cells are also the wellspring of cancer. A research group in Madrid reports that stem cells taken from adults can turn cancerous if they are allowed to multiply for too long outside the body. A better understanding of the mechanism whereby undifferentiated stem cells become differentiated may yield information on how cancer arises through abnormal cell division and differentiation. Cancer stem cells might continually replenish tumors. Indeed, this might explain why treatments that can shrink tumors don't always cure the disease. Perhaps the treatments are less effective at killing the cancer stem cells. Cancer stem cells use a variety of biochemical pathways to survive. Identification of the biochemical switches they use to reproduce could provide the target for a silver bullet drug. In 2005, McCulloch and Till received the Albert Lasker Award for Basic Medical Research. In an article published a month later, they paid tribute to the vastly undercredited elements of both luck and astute observation. We weren't deliberately seeking such cells, but thanks to a felicitous observation, we did stumble upon them. Our experience provides yet another case study of both the value of fundamental research and the importance of serendipity in scientific research. Reflecting on his work in an interview in 2006, McCulloch said he had always thought that the idea of the scientific process was overblown. He knows that typically a successful scientist may start with an experimental design, but then makes an unexpected observation that leads a prepared mind to follow a chance event.